Hello, my name is Avery Pearson, and I am a recent graduate of the College of Worcester. Today I'm going to be talking to you about my senior thesis research. So to start off, we'll just do some background on plants. So as you all know, plants are incredibly diverse, and in many ways, including their reproductive modes. The largest group are hermaphroditic, about 90% of angiosperms, but most of these are in fact self-incompatible, about 60%. Because of this, many plants rely on a vast array of pollinators to assist with reproduction. These pollinators can take a wide variety of forms. There are over 20,000 different species of bees worldwide. While you might be most familiar with honeybees, most of our bees are in fact solitary, about 85% of our total species worldwide, including most of our native species in North America. These solitary bees are often ground nesting and do not live in colonies or have specialized duties and don't have overlapping generations like our social bee friends. Pollinators can be split into two major categories. The first are our specialists. So specialists only visit one or a couple different species of plants when they're gathering pollen, and because of this, they're oftentimes better at floral handling. So in my system of Echinacea angustifolia, our specialist species is Andrena helianthiformis. So you can see our little cartoon here. As opposed to specialists, our other category are the generalists. So generalists visit a wide variety of different plants, and because of this, they oftentimes are not as skilled at floral handling because there's lots of different plants to visit. Some of our generalist species in my system are Agacorella, so that little green bee, our Halictus, which is the large gray bee, and the black and yellow bee there, our Megachile. So because our bees are helping to move pollen, they're influencing our plant's fitness. So fitness is essentially how effectively an organism survives and reproduces to pass on its genes to the next generation. Specifically, I'm going to be focusing on reproductive fitness, which is comprised of two major parts, female fitness and male fitness. So in plants, female fitness is essentially how many ovules are successfully fertilized, or what's the seed set. Male fitness, on the other hand, is who donated the pollen. So as you can see here, based on how we are approaching these, it's easier to measure female fitness versus male fitness. Female fitness, you just have to count the number of seeds that are successfully set, whereas male fitness, you have to go back and trace where a single grain of pollen came from. So because of this, there's been a lot more research done with female fitness in plants compared to male fitness, so understanding both is key if we want to understand reproductive fitness as a whole. My study species is Echinacea angustifolia, which is a prairie perennial and native to the Midwest and western parts of the United States and parts of Canada. It is hermaphroditic and genetically self-incompatible, and is also a member of the aster family. So the aster family is one of our largest angiosperm families and is characterized by its head inflorescence. So this means even though it might look like one flower, it's actually composed of many tiny little florets. So if you look down here in the corner at our picture of Echinacea, we have a red arrow and that's pointing to this little yellow dot. So that is actually one floret on the head of Echinacea, and it is in its male phase, it's presenting pollen. And then if you look at our blue arrow, it's pointing at this little Y-shaped structure, which is actually a receptive style of Echinacea. So this floret is in its female phase. In Echinacea, it is uniovulate, which means that for each floret, it corresponds with one seed. Echinacea has at least 26 different species of pollinators identified so far, and most of these are native solitary bees. Some previous research that's been done in this system was investigating the impact of different pollinators on female fitness of Echinacea. So in this study, they looked at the number of receptive styles at the time of a pollinator visit, and then they measured the proportion of styles shriveled after the visit. So in Echinacea, style shriveling has been shown to be directly correlated with seed set. So the more styles that are shriveled, the more seeds that are set. So here, each pollinator is represented by a different color and shape. So you can see here, there's lots of different pollinators, and lots of them are clumped down at the bottom. But our one major trend appears to be that the gray dots, which represent our specialist pollinator Andrena, are much higher up than the rest of the pollinators on average. So this shows that per visit, Andrena results in significantly more style shriveling, so it is a quote-unquote good pollinator from a female fitness perspective. I wanted to build on the findings from this study by investigating how our different pollinators of Echinacea influence its male fitness. So in order to do this, I had a two-part study, and the first part was in the field. So our field site is out in west central Minnesota, where we have an experimental plot of Echinacea individuals. So though Echinacea is hermaphroditic, for the purposes of this experiment, we made it functionally dioecious. So we had two categories. We had paternal individuals, which were our pollen donors, and maternal individuals, which were the individuals we were collecting seeds from and assessing the offspring. So our paternal individuals were restricted in pollinator visitation. So say an Andrena came to visit this paternal individual. Then for the rest of the day, we only allowed other Andrenas to come visit, and we shooed the rest of the bees away. Similarly, if a green bee came to visit this individual, we'd only allow other green bees to come visit it for the rest of the day. On the other hand, our maternal individuals were not restricted in the type of pollinator that was able to visit. Though they were still presenting pollen and it could be moved around, we were worried that if we emasculated the maternal individuals, the bees wouldn't have a reward to come visit. Outside of experimental times, we used these pollinator exclusion bags. You can look up in the top corner to see a picture of that, to make sure that pollen was only moving during experimental times. This field experiment took place over 5 days, and I'm going to be looking at just the results of 2 days of this experiment. So after the field season was over, we took all the seeds from the maternal individuals and shipped them back to Ohio, where we germinated them, extracted DNA, and we performed microsatellite analysis on these offspring and the parentals individuals from the field. After we ran the PCRs with the microsatellites, we sent off these PCRs to a genotype facility which performed capillary electrophoresis. Then we got back these charts. So you can look down here, this graph with the different fragments, which we analyzed, and then we ran these results through a package in R called MasterBase, which assesses for most likely paternity. So essentially, it is able to match up offspring with a most likely paternal candidate based on how well their microsatellite results match up. And since each paternal individual corresponds with a specific pollinator, we can trace back which pollinator moved the pollen to result in this offspring. Looking at our results, in terms of field visitation, we found that there wasn't a huge difference in the average number of visits for each pollinator group to a paternal plant. So they're all around between 1 and 3 for their average visitation. So as you can see, our 4 categories here that had sufficient number of visits for us to analyze were A and D, which is Andrena helianthiformis. That was our specialist pollinator. We have Halictus, and then small black bee, and open. So as you can see, the first 3 groups here, Andrena, Halictus, and SBB, all correspond with an individual pollinator group. So our open individuals were actually our maternal individuals. So since they were still presenting pollen, the pollen from the maternal individuals still could have resulted in some of our offspring. So that is what this category represents. Moving on to siring success. So we ran our results through MasterBase to pair up offspring and their most likely paternal candidate, but we can also run a model in MasterBase using different predicting factors to see how well different factors predict for paternity. So in our model, we used our different pollinator groups and the number of anthers presenting on a given day as our predicting factors. So we got an output value that essentially represents the proportion of offspring sired by that group. So if we look here, Andrena has a value of about 0.11, so it best means that it sired about 11% of our offspring, whereas Halictus was 6%, SBB around 26%, and open around 12%. So our SBB had the highest siren success rate. So as I mentioned, we also ran this model using number of anthers as a predicting factor, but the coefficient that we got as an output for this was only about 0.001, so it wasn't a significant predicting factor. But what's perhaps more intriguing is that the SBB group was the highest in siren success rate. So we might have thought that our specialist pollinator, Andrena, would have the highest siren success rate because it had the greatest impact on female fitness per the Page et al. Study that I mentioned earlier. However, one possible reason that the SBB siren success was higher is because, as their name suggests, the small black bees are small in body size compared to our other groups here, Halictus and Andrena, both of which are mid-sized bees. So perhaps by restricting pollinator visitation, we helped reduce interspecific competition for our SBBs. Because of their smaller body size, perhaps they're more likely to get outcompeted by the larger bees when visitation is not restricted. And by restricting visitation, the SBBs could visit the flowers more and pick up more pollen and drop off more pollen, which could lead to higher siren success. The results of this study are interesting in terms of what they mean for our understanding of reproductive fitness of Echinacea. So the Page et al. Study shows us that Andrena helianthiformis has the greatest impact on female fitness, whereas the results here show us that the SBBs have the greatest impact on male fitness. And so as we know, reproductive fitness is a combination of female fitness and male fitness, so we need to take both of those findings together to understand reproductive fitness as a whole. So looking ahead, I would like to run the same experiment with the rest of the offspring, so the offspring that resulted from the other 3 days of the field experiment. So actually, I would like to perform further analysis with different predicting factors. So as I mentioned, Master Bayes is able to run a predictive model based on the input of different variables. Here we used our pollinator groups and the number of anthers presenting on a given day, but we were not able to factor visitation into this model, which would be interesting to look at moving forward. Additionally, I'd like to investigate other measures of male fitness. Our lab is looking for other ways to measure male fitness, so if you have any suggestions, please send them our way. I did this research as part of the Echinacea project, which uses Echinacea angustifolia as a model organism for prairie conservation of plants. So the results here show that we need to broadly conserve the pollinator community, and help facilitate a diverse pollinator community in order to help Echinacea be most successful. Though the results here showed that the SBBs had the greatest impact on male fitness, previous studies have shown that Andrena had the greatest impact on female fitness. Therefore, we need to conserve both these species in order to help boost reproductive fitness as a whole for Echinacea. If you have any questions or comments, please reach out. I'd love to hear from you. Thanks again for stopping by, and have a great rest of your day.